Francis Crick proposed the adaptor hypothesis, which states that small RNA molecules would be adapters that could receive a specific amino acid and then interact with mRNA. This transfer of information is turning one kind of information into another to communicate it. This is similar to an adapter, which is a device that is used to connect two things that cannot be connected directly in order to transmit information. An analogy of this would be when you try to play a song from your phone in your car, you cannot plug the phone right into the aux cord. And since your phone won't directly connect to the aux cord, you need an adapter. In this case, the phone is the mRNA, the song is the DNA, because it is the message that is being transcribed and then translated. This part of the phone is the codon, and this part of the adapter is the anticodon, because it is the one found on one end of the tRNA. The dongle is the adapter, or tRNA, and the aux cord is the protein. When the song starts playing, it is like a gene being expressed. So let's take a look at the central dogma. This is the process by which the information in DNA are converted to a functional product, which is proteins. The DNA is transcribed into mRNA, and then the mRNA is translated into the amino acid chain, or protein. Amino acids have side chains that are hydrophobic and hydrophilic, but nucleic acids of mRNA are composed of nucleotides that are specifically for hydrogen bonding. This makes it so that nucleic acids are able to recognize one another, but not amino acids. Francis Crick proposed that there was an adapter that provided the link between the codons of the mRNA and the amino acids for which they coded. His adapter hypothesis effectively predicted the existence of tRNA in this process. He predicted that the tRNA would be the adapter that could identify the codons on the mRNA by base pairing, which is shown here. The anticodons are complementary to the codons of the mRNA. tRNA is composed of an amino acid binding site on its 3' end and an anticodon on its 5' end. Ribosomes are the location of translation. Ribosomes are composed of rRNA and proteins. The ribosomal unit binds to the 5' end of mRNA. The genetic code of mRNA is in the form of codons. These are read with our tRNA, which allows us to pair these right here. These adapters could then incorporate this information into the amino acid chain. So what would happen if tRNA cannot act as an adapter? An alteration that prevents tRNA from acting as an adapter that follows the genetic code is certain mutations. These tRNA could be charged with the incorrect amino acid due to a problem in the enzyme aminoacyl synthetase. There could be a mutation of the anticodon length in the tRNA, so the anticodon could not bind to the codon and start translation. The consequences that may occur to the cell could be that the organism could die and certain essential functions of the cell may not work. Also, this could lead to a change in the biological processes of the organism. In conclusion, tRNA is very important in cell function.